It's a great honor to be participating in this meeting, which I found fantastically interesting. Several of us in this meeting have had the privilege of living in large part of a golden age of biology, which in the last 60 years has achieved an amazing progress in our knowledge of the living species that populate this common home, as the Pope Francisco calls it. In these 60 years, we have moved from not knowing how genetic information could result in the beautiful biodiversity that we find on this blue planet. We have really seen a whole revolution of knowledge. And we're now at the present position in which we can virtually guide evolution of living beings on Earth. Molecular biology and biotechnology are key sciences which we will have to use to solve the very serious threat that we're experiencing today, and as a result of human action and also of lack of human action. In the following slides, this one. This is too small for you to read, I can see, but it doesn't matter because I'm not going to go into detail. It's just, I like to make a list of great achievements during the last 60 years, starting from the discovery that DNA was the genetic material to the present time. And in the present time, there I have 2015, in which in March of this year, in the Science Journal, there was a call for a moratorium on using a new technology called CRISPR-Cas9, which is a very powerful technology and very inexpensive and rapid technology for engineering genomes. And this call, which is signed by a number of very distinguished scientists, two of them Nobel Prize winners, call for this moratorium to use this technique in the human germline until more is known about the difficulties and the possible risks of using this. And as a matter of fact, in a few days' time, in the beginning of December, there will be a meeting in Washington called by the U.S. National Academy of Sciences, the National Academy of Medicine, the Royal Society, and the Science Council of Japan, calling to discuss whether this moratorium can be agreed on. I think this is an important consideration that maybe this academy can consider, because obviously, as has been already said here several times, ethics is a key factor that we have to consider to transmit to the children of the 21st century. The rapidity of the progress of knowledge of biology in the last 60 years has been phenomenal. And for those of us in this area, it has been very, very difficult task to keep up with the progress of these sciences in our own special disciplines. We can understand, therefore, that it is impossible, the impossible task that the secondary school biology teachers are expected to accomplish in preparing their students in all of biology before the students enter the universities. We that work in the universities have of many countries find that students that register to start undergraduate work lack the solid understanding of some of the fundamental concepts of biology and other related sciences, and even more seriously, also lack the practice of using the critical thinking that is the basis of science. This situation is related to the fact that in many countries, only small numbers of students are taking the option of studying sciences as their life careers. This contrasts with the fact that we're clearly now in a period in which knowledge and the generation of knowledge through science are the main ways that humanity has to overcome the grand challenges that we face in the world. It is clear, therefore, that we must try very hard to improve science education and practice in all the levels prior to the university. One of the most serious shortcomings of science education in most Latin American countries is that it mainly concentrates on theoretical lectures that require students to memorize long lists of strange names and formulas and equations, and that the students cannot relate this to their everyday experience. Very little of the science education is dedicated to doing science, which for us means doing experiments. To learn science, one must do science. It is common practice to give us as an excuse for not doing experiments in primary and secondary schools the reason that laboratories and their equipment are too expensive for our schools. The recent introduction of inquiry-based science education, IBSE, modules designed to teach basic concepts of primary school science are with extremely simple and inexpensive materials that allow children to do hands-on experiments has completely eliminated this excuse and has demonstrated that in many countries that science classes can be entertaining and that children enjoy very much learning about themselves and about the surrounding universe by doing experiments. Secondary school science, especially in the last years, just prior to the entrance to the university, is more sophisticated and the equipment necessary to do experiments is more expensive. The other important consideration that hinders the performance of experiments at that level is that secondary school teachers of science receive their initial training in institutions that often do not carry out much original scientific research. For that reason, their scientific training includes very little experimental work, and they lack the expertise in designing the experimental protocols and in the handling of scientific equipment. The Latin American Network of Biological Sciences, called RELAB, started in 1975 as a regional project of UNDP and UNESCO, which had the main objective of strengthening postgraduate training in biological sciences in the Latin American countries. This project was responsible for a large series of training courses that included teaching of the theory and practice of the new techniques of molecular biology to postgraduate students. During the late 90s, we started organizing theoretical and practical courses to teach secondary biology teachers some of the fundamental aspects of molecular biology. In 2001, I had the honor of participating in the meeting organized by the Pontifical Academy of Sciences called the Challenges for Science Education for the 21st Century. In that memorable meeting, I presented a lecture titled The Problems and Promises of Science Education in Chile. In that meeting, that was published in a book of the Pontifical Academy of Sciences. There is the book, and there I include page 91, which is of that book, in which I talk about the training of teachers in molecular biology. But one thing I did not say is that although the teachers liked very much the courses that we gave them as professional development, they were a little frustrated because they could not do the most interesting and exciting part of the course, which was doing the experiments. So I include the last sentence there that is underlined. It says, we have the project of developing a mobile DNA laboratory that can visit schools and let the students carry out experiments of genetic engineering. My presentation today demonstrates how slowly we have moved to implement this idea. In 2011, RELAB applied to the Wellcome Trust, which has been mentioned already here this morning in the United Kingdom, which at that time had a program to link the life sciences to society. In this project, it was presented by 3 countries: Mexico with the University UNAM, the main state university, Costa Rica by the University of Costa Rica, and Chile by the University of Chile. The project proposed to conduct training, theoretical and practical courses of molecular biology and genomics in each of these 3 countries. The secondary school teachers that approved these courses could request the university in that country to send a portable laboratory with all the equipment and materials necessary for a group of 30 secondary school students to do the same experiments that the teachers had done in the university. In 2012, the Wellcome Trust approved this project, providing RELAB with approximately $48,000. This project had the general objective of contributing to the improvement of biology teaching in molecular biology and genomics through a theoretical and practical professional development of secondary school biology teachers, and the courses were designed to concentrate on only 4 fundamental concepts. And this is quite important because one of the big problems is that the curriculum, most of the curriculum for schools that the ministries of education prepare are too broad, too many subjects, and they are not concentrating on fundamental concepts of teaching. To show you how fundamental those concepts, those few concepts were, I will read them. One of them is the genetic identity of species and individuals is determined by the information present in the structure of the DNA of the species and individuals. Phenotype comes from genotype. The second one is that most cells of a multicellular organism contain the same genetic information as the total organism, and yet cells have different morphology and function and express different proteins. This is achieved through regulation of the expression of genes. The third concept was genetic code, the genetic code that is used to translate the DNA nucleotide information to generate proteins present in the cells is practically universal for all organisms. And the mechanism for translating that information from nucleic acids to proteins is very similar. These are essential facts for genetic engineering. That's what made genetic engineering possible. The fourth concept was that Darwinian evolution can be observed at the molecular level through the study of sequences of genes and their corresponding protein products in different related species. Molecular genomics can provide information regarding the time scale in which the different phylogenetic branches emerge from the evolutionary process. Well, we agreed that we should concentrate on this, and we decided on a series of theoretical lectures to illustrate those 4 fundamental concepts, and in experiments that could support the validity of those concepts. Okay, the courses for the teachers were given in 5 concentrated days with 8 theoretical lectures and 4 experiments. The lectures were given in the mornings, the 4 experiments done in the afternoons, and the 5th day we dedicated to evaluating the course, to discussing the applications of molecular biology and agricultural medicine and preservation of the environment. And so this was a condensed, very short course because teachers cannot attend for long periods of time training courses while they're keeping their duties going. So in Chile, the portable laboratory is really portable. You see, this is a portable laboratory that has all the equipment that will be required for the experiments. It can be put in 2 suitcases with wheels that can go in any car, in the back of any car. So it's very easy to transport this equipment. I was going to go into the details of the experiments, but I think it's going to be too long, and I see that the time is running very, very fast. So I will skip this. Maybe if somebody has a question at the end in the discussion period, we can do it. So I will, but this is a very important point, that in Chile, where the laboratory, the portable laboratory, is not going by itself. It's accompanied by 2 doctoral students in molecular biology, young persons that are doing these techniques every day in their thesis work, and they know them very well, and they can help the teacher in the performance of these experiments with the students. This has been a key factor for the success of this, because it not only gives the teachers this support, but also it provides a dialogue between very young people, young scientists like the doctoral students with the high school students. So it's a very positive aspect of the project. This goes on to tell you about the different experiments. For instance, in this one, bacteria transformed with the green fluorescent protein that can show that the genetic code is practically universal, and that genes are turned on and turned off. Is a very nice experiment. Also the experiments with the sequence analysis of genes and proteins is shown here, but I'm not going to go into that detail. So we have this project now for 3 years in an experimental pilot project in Chile. We have conducted 3 courses for biology teachers and trained around 70 biology teachers in these techniques. And we have visited in the years 2013 to 2015, 27 schools. We have been able to visit 27 schools, most of them, 26 of them, in the metropolitan area of Santiago, only 1 outside of Santiago. And we have had the opportunity for 750 students to do experiments that involve sophisticated experiments of PCR, of genetic transformation of cells, many different aspects. So if you divide the cost of the portable laboratory, which was $10,000, the portable laboratory, all the equipment would cost $10,000, but 750 students that we have already performed experiments is around $13 per student, which is less than providing them with a modern textbook of molecular biology, and this is a much more effective way. And the laboratories are not destroyed, they're still there. We will still use those in the next 2 or 3 years. So the price of per student is gonna keep coming down in this experiment. Okay, so there are many opinions of the doctoral students and of the teachers that we have collected. We have in a web page more than 400 opinions of secondary school students saying how exciting it was for them to do experiments that they could read in the papers about doing, and that now they were doing it in their own school room. The school room, many of those schools, they only had to have electricity, access to water, and not too much more than that. And obviously for all the schools in Chile just about have computers online, so they could do the experiments to go into bioinformatics experiments. So, but one of them, for instance, I would maybe read one of the opinions of a graduate student. Well, I'm sorry, I got, but I will give you, yes. I want to add, one of the graduate students says, I want to add that this initiative, I would like to suggest that the participation of this project not only be limited to students, because I have graduated, I got my doctorate degree, and I want to continue going to the schools with the laboratories to teach the children. They found it very exciting and want to continue. And the teachers say that increased interest in science is one of the results. They also consider that many of their students are changing their minds of what to choose, what career to choose. They now see that science can be done in Chile, which was a very negative aspect. 85% of the 750 students that have answered this question said they had never seen in person a scientist or a graduate student of science. So it was impossible. They had no role models around them to guide them to choose a career in science. And so we're expanding this, project. This is going on in the beginning, 3 countries. Uruguay has joined in, and now this year, next year, 2016, Brazil, Panama, and Peru are joining in. And we're also escalating. We're developing new modules. We're conducting workshops of scientists and educators to design new modules in other chapters of biology, but we would like to go into other experimental sciences. So it could be a very broad project. Maybe in the general discussion, because my time is up, I can go into more details. Thank you.
Thank you very much, Professor Allende.